looking at chapter 5 now. So tissues. So here are your learning outcomes again, as usual. So let's get into this. So we have 4 main types of tissue, and connective and epithelial, muscle, and nervous. All right, so the way we usually do this is we start off with epithelial, so that's what I'll be doing, but we're going to lead into that a little bit, and you'll see that this chapter is mostly focused on connective and epithelial tissue, and I'm just introducing muscle and nervous, and that's because we're going to spend a lot of time studying those 2 types of tissues later on in the semester. So we'll talk more about the specifics at that time. And some of it, of course, you'll hear again from what I'm going to talk about here. Now, all of the tissues are derived from 3 what we call germ layers in the embryo. All right, so those 3 layers are the endoderm, mesoderm, and ectoderm. Okay, so when we're talking about a tissue, all right, so, and I mean, it sounds weird that they're, but ultimately, that the whole embryo or the whole person is derived from a single cell, right? So, and there's this whole developmental program that reassigns and shifts cells, and I'm going to talk to you about little things or different ideas that hopefully will help you appreciate how that works, all right? Because ultimately, what is a tissue? It's just, you can see it, the standard definition on the first page of the notes, right? It's this group of cells with a common embryonic origin that, while it could be very diverse in what it looks like, it's going to carry out a specific or specialized activity, okay? So ultimately, though, and there are different types of ways that organisms are formed. Like some types of organisms only have 2 germ layers, all right? But we have 3. So humans have 3, and they're called the endoderm, the mesoderm, and the ectoderm. And you can sort of see what is derived from those different types of tissues. Okay, so mucous membranes, I'm going to talk about what those are again, digestive glands, and then mesoderm, muscle, bone, and blood, and the epidermis comes from the ectoderm, as well as the nervous system also comes from that. So it sounds kind of weird, your skin and your nervous system, but they are derived from the same germ layer. And you can see also on page 1 the standard definitions of each of the 4 tissue types, right? And we're going to get more into that. But if you look at it, connective tissue in general is protection and support. It's going to bind organs together. It's going to be involved in immunity, storage of energy, extracellular matrix, so the matrix that's outside of cells. You're going to have more matrix than you have cells. And in general, it's vascularized. Epithelial tissues are going to cover surfaces and line hollows and ducts, form glands, connections between the internal and the external environment. And in this case, unlike connective tissue, there's more cells than there is matrix or extracellular matrix. And in general, a lot of epithelial tissues are not well vascularized, meaning that they have a poor blood supply, whereas the connective tissues are well vascularized, having a good blood supply. So that, and for that reason, they work together. Now, muscle and nervous tissue are both excitable, and explain what that means. Muscle tissue, of course, is the ability to contract. It generates force and heat, and there's 3 different types. We'll talk about that. So skeletal, cardiac, smooth, and nervous tissue is going to detect and respond to external signals, generates action potentials, brain, spinal cord, nerves, and there's accessory cells, or what we call glial cells, associated with that. So we'll get back to that, all right? So all of those tissues are derived from these 3 germ layers, all right? And we don't automatically have those 3 germ layers, right? So there's a certain program of development that takes place from your single cell up until this stage called gastrulation, right? Gastrulation. And it's this point, you can see it here, you've got your 3 germ layers, all right? So the mesoderm is always in the middle. And then you can see that there's the ectoderm, and then the endoderm is always the one is more is deeper in the embryo. Okay, and actually you can see it surrounds the yolk sac. All right, so it's kind of interesting the way that works. But it develops from that single cell, and you can see that there's a certain stage. Just day 4, you've got this big ball of cells, and this one here is called the morula. You're going to learn more about this in AMP2. Right at the end of the semester, there's a little short bit on embryogenesis. So you're going to look at this, and you'll look at chicken embryos in lab. And if and here you can see exactly this video here. It's really quite nice because it animates how this gastrulation takes place. And there's a whole course called developmental biology in which you can study the full formation of an individual and how they develop to adulthood. It's really an interesting course. I actually got to teach it once. It was really interesting because I'm not an embryologist, so I was able to learn a lot as I was teaching the students, and it was really quite an enjoyable. It was tough, I'll put it that way. It was tough, but it was an enjoyable experience because I did learn a lot doing that. This is an aspect of anatomy and physiology I hadn't really explored yet. So if you look on my notes on page 2, you'll see a little bit more detail about what types of things are coming from each of these. And hopefully, like, when you think of mesoderm, you can sort of think of the middle, right? So, the thing that sort of causes things to function and stuff. So in that way, you think of muscle, bone, and blood. That's sort of the essence of making things work. And then ectoderm, I think it makes sense because ecto usually means outside of. So that would be epidermis and that kind of stuff. And just remember, nervous comes with that. And then the endoderm is going to be these sort of secretory things, right? So mucous membranes. Mucous membranes include your digestive and your respiratory system and the glands associated with that. And you might not be thinking about digestive and respiratory as being a secretory membrane, but the membranes there that line those systems, they actually produce mucus. So they are sort of secretory, and they also have glands as well. So this is something you just have to sort of learn, but I'm trying to give you some ways of trying to separate which types of tissues come from which germ layer, all right? Here you can look at it in a little bit more detail. You can see here, the implanted embryo, and you can see the orientation of these different, tissues when you're looking at it here. So your ectoderm and your mesoderm always is the middle, and then of course the endoderm right there. And in the endoderm, it's always around the yolk sac. So that sort of gives you that sort of perspective a little bit. Here they don't really differentiate between the yolk sac and the endoderm, but if you look back here, you can sort of see how that works. And the really important thing to understand here is that these cells are moving. Like, it's really, really fascinating. And later on, when we get into different types of adhesions, I'll sort of explain a little bit about an adhesion type of junction that allows cells to move, and other ones that keep cells in place. All right. But remember, the cells here are very motile, all right, in what they're doing. And that's why it's nice to watch this little video here. It gives you a little bit of perspective on the movement of the cells, all right? Okay. Now, you're going to spend a lot of time looking at histology in lab, and I'm hoping you'll get lots of time to do that. Otherwise, you're going to have to do it online looking at pictures, but it's really not quite the same thing. To really actually look at the tissues and appreciate them. And for me, I don't really, like, I took it was really hard course for me because it was a lot of work. It wasn't that it was, mentally challenging. It just was a lot of work to learn. I took histology, like a whole semester, and all I did was histology. And I learned an incredible amount there. But then when I started, after I'd done my grad work, I was doing my first postdoc. That's when I really started looking at tissues. And it was really, really, it really gives you a different appreciation of what goes on with cells when you see them in the context of everything else that's going on. And I'm going to be talking about how this context is so important. So this is why it's nice to study your slides, and you'll appreciate that your tissue isn't just this. the slide that you're looking at from whatever organ it is has all kinds of stuff going on. And you don't get that perspective if you just look at specialized pictures on the internet, because it hones right into what you're supposed to see. But if you actually spend time looking at your histology, your slides in lab, you'll learn how to spot those different types of tissues in the context of the rest of the organ, which is really a really important way to understand how the body works, right? Now, in order to make those slides, there's 3 steps that are done. So first of all, they have the fresh tissue, and it has to be cut into like a manageable size that can be looked at, right? So they can't just take a whole liver and then look at it. And then they fix it. So there's a fixation is basically, it kills the tissue, but it's supposed to preserve it in a way that's representative of what it was in the living state. And you'll see some of the slides in lab are really lousy because they did really poor fixation. And other ones are lousy because it's poor sectioning, which is the next step. But and there's different types of fixation that can be used, right? There's a whole bunch of different fixatives. I know most of the time people just think of, formaldehyde, because when you're in school, you used to get your preserved, cats or whatever. They can't actually use formaldehyde anymore. It's usually paraformaldehyde or something, alcohol or whatever. But there's different ways of fixing the tissues depending on what you want to do at the end, right? And then sectioning. So they're going to be sectioned in different orientations depending on what you're looking at, okay? So here you can see the different types of sectioning, so longitudinal cross sections and oblique. I'd imagine if you're looking at a pathological sample, it's going to be largely oblique because they're going to try and get into the part that they're really interested in to look for the pathology. And then they're going to be stained, and because tissue, you can't really see much. So there's going to be stained with different dyes to highlight what is desired by the end user, okay? One of the most common types of stains, a very generic stain, is called H&E, so that stands for hematoxylin and eosin. And most of the slides in the lab are H&E stains, all right? There's another way of doing certain tissues and fixing, actually you don't fix it, you flash freeze them. So it's called frozen sections, and or the cryopreserved, and then they're cut right away and dehydrated. So that's another way of doing it, and it's much faster, but it's not always the most desirable, but sometimes it is the more desirable depending on what kind of staining you're going to do. So, but traditionally, things are fixed in formalin, so that's like formaldehyde with some buffer in it. They're sectioned and then they're stained with H&E. So that's the standard process. And this video that I have on this page is a little long, but it's really interesting. And it really shows you the process. And if you're someone who's heading to the clinic as a nurse or whatever it is, it's really worth watching, even if this is the only time you see it done, so that you understand what are going to be done with your patients' biopsies and what in the process that's going on. It's not something that you easily forget once you see it done. So I encourage you to watch it. It will be useful to you as you move forward in your career. Even if you don't end up doing it, you can actually get a job doing this. It's actually a very good job. But you'll see the technician doing that in the video. But it's really, it's really interesting, and I think it will be helpful to you to appreciate what's involved, even if you don't end up doing it. But then you'll understand what's happening. Right, and you won't forget. You'll see it, and it's something that will stay with you. So that's why I encourage you to watch it. Okay, now we're going to start off with epithelia. All right, so there are lots of different epithelia in the body. All right, so they're found all over the place, and in general, you could think of them as linings, surfaces, and coverings. All right. And they're classified in different ways, all right? So they're classified first by the number of layers and then by the shape of the cells themselves, all right? So these are the basic ideas here. So simple just means that you have one layer of cells, and stratified is the opposite. You have more than one layer of cells. And you're going to see there's an example called pseudo-stratified. And anything pseudo is pretending to be something that it's not, all right? So what's happening here is that you have basically a simple situation, but it looks like it's stratified. And that's because the nuclei, which are these purple things, all right, these purple guys, they're in different places. So it gives the impression that it's stratified, all right? But if you would really look at it, you'd see all the cells are attached to the substratum. I'm going to talk about what this is called. This is the basement membrane, all right? I'm going to get into that in a little bit. Now, the other way to classify the cells is by their shape. So there's a squamous, which sort of looks like cobblestone. Now, if you look at it from the top, it looks like cobblestones, all right? So irregular shaped cells. Now, in your lab, you're usually not going to see it from the top, okay? You're going to see it from the side. So they would be very flattened cells, all right? You're going to usually see this from the side in the section, all right? It's very difficult to slice this and see it from the top, all right? Cuboidal, the cells are like little cubes, and columnar, they're like columns. So they're taller than they are wide, all right? And then, so when you name your epithelium, you use both terms. So you can say simple squamous, or simple cuboidal, or stratified squamous, or stratified cuboidal, all right? So when you're naming an epithelium in lab, use both terms. Don't just say pseudostratified, right? You know, say simple stratified. Usually it's going to be ciliated, you know, and you'll see that when you're in lab, all right? Or a simple columnar. Don't just say columnar, all right? Proper way is to say simple columnar. Most columnar tend to be simple, all right? But when you're learning, make sure you use both terms, okay? All right. All right, and then here's some examples about where you can find these different types of epithelia. So you notice here you've got simple squamous, simple cuboidal, simple columnar, pseudo-stratified columnar, stratified squamous, stratified cuboidal, stratified columnar, transitional. That's the one I haven't mentioned yet, so I'm going to talk about that now, all right? And you can see here, you know, types of functions. You'll see if you have more than one layer, it tends to be somehow protective, right? You'll see the protective under each of these, all right? Whereas if you have a simple, usually that means that things can get through. So there's going to be secretion or an absorption happening here, all right? In general. Now, transitional epithelium, that's a really weird beast, okay? But this is where you have, and it's usually in the urinary system, so it's lining the bladder, the urethra, and the ureters. And the thing is now, this is expandable, all right? It allows stretching and expanding of these organs. And that's usually where you see this. And it looks like a hot mess when you see it histologically. And I think I don't have a picture of it here. But if you look in lab, you'll see that there is a slide of bladder. And I suggest you look at it. You'll see what I mean, right? So it does look like I said, a hot mess. Because you'll see that the cells have all these weird shapes, and there's, you know, all kinds of different layers. So it doesn't really look like any of the other ones, okay? Okay, now, I'm just seeing what I had on my notes here about transitional, yes. So it allows the organs to expand and contract. The number of layers are important in that process, and the cells can change shape, okay? Now, epithelial versus connective tissue. So there's this relationship between these two, and I sort of alluded to that already, because epithelials, there's lots of cells there, but in general, they are avascular, so they don't have very much of a blood supply. So they need their blood supply from something, and they also need to be supported because they're like a sheet of cells, right? So they're always supported by connective tissue. So connective tissue, there's lots of different types of fibers there, and they're made by fibroblasts, all right? And there's a good blood supply. So they work well together, and you'll see the consequences of that specifically in skin as those cells get further and further away from the blood supply, right? So we'll talk about that shortly, and I think in the next chapter, all right? So these two types of tissues are usually found in association with each other, specifically epithelial. They'll always have to have some kind of connective tissue associated with them, okay? Now, if you look in my notes, I have sort of a picture of what to expect here. So you'll see that the epithelial cells are at some sort of a surface, all right? And then underneath, there's going to be this structure called the basement membrane, and then the connective tissue is going to be underneath, and well, deep to that. And on the other side, on the top, on the lumen, there's going to be the outside or a cavity or a tube or space of some kind on the other side. So there's connective tissue on one side of the epithelium and then like some sort of a space on the other side, right? Now, the epithelium is going to be connected to that connective tissue or basement membrane with specialized junctions, and I'm going to talk about those, right? So here, if you look at this a little bit more carefully, so here's a pseudo-stratified columnar epithelium, and you can see that it's ciliated. So I'm going to go back here. So you've got your pseudostratified columnar, right? And it's got these structures called cilia on there. I'm going to get into those shortly. You can see that's another feature here. And notice now, that you've got them attached to this basement membrane, all right? And this basement membrane is made up of a few layers, okay? So there's the basal lamina itself, which is the part to which the epithelium is actually attached. And then below that, there's the lamina densa, okay? I mean the reticular lamina, sorry. So there's the basal lamina and the reticular lamina. And this basal lamina is made up of two layers. So lamina lucida and lamina densa, and then underneath the reticular lamina. And all together, those make up the basement membrane. All right. So this sounds really complicated. All right. I know that, and I'm not going to test you on that. I want you to know that there's a basement membrane to which the epithelial cells are attached. That's the important part here. This is more for you to understand and to appreciate the complexities of what's happening here. All right, so here you can see that this basement membrane isn't just a generic layer of connective tissue. It's very specific structure to it. And you'll see here that there's actually very specific types of proteins in each layer, right? So you've probably heard of collagen before, but you might not realize that there's different forms of collagen, all right? And so here you can see in the lamina reticularis, or the reticular lamina, you've got collagens 1, 3, and 5. And then the lamina densa, you can see a collagen 4, right? And so on, right? And laminins, you can see there's different types as well, all right? This is more for you to just appreciate the detail and the complexity of what's happening here. What do I expect you to know? Well, like I said, when we first started this course, you're going to learn things as you go. And, this might be very important depending on what you end up doing later. That's why I want to expose that to you, you to that. But right now, just know that there is this basement membrane that the epithelium is attached to, that mediates its attachment to the connective tissue. And understand that, there's a system here, and there's different types of proteins involved in that basement membrane, okay? And the next thing I'm going to show you is how those epithelial cells are attached to that basement membrane and to each other, all right? Because these epithelia, if they're going to make coverings and linings, they have to, be able to hold on to each other as well as to this basement membrane. So we're going to talk a little bit about that, okay? Now, other little things. In lab, you're going to hopefully spend some time learning to identify these, right? So here's just some slides to help you appreciate that. So here you can see I have an arrow pointed to a simple cuboidal epithelium, and it's been colorized so you can see it a little bit better, right? Your tissue samples in lab aren't going to give you that kind of hint, all right? But you can see that these are sort of cuboidal cells. Remember, you're not going to be sectioning each cell in exactly the same place, okay? But this is not bad. You can see that this is a pretty good cut through this epithelium. Whereas here you have a simple squamous, right, up here. So you can see they're very flat. There's one layer. And here again, you have simple cuboidal again, all right? And this is in the same tissue slab, on the same slide, okay? This is connective tissue in between. There's some fibroblasts in here, all right? But you'll be able to identify these. Here, again, you have a simple pseudo-stratified columnar epithelium and simple columnar epithelium and a pseudo-stratified columnar epithelium, right? So here are the differences. So here you can see that this pseudostratified columnar epithelium actually has the cilia on it, all right? And very often it does. There's another type of cell here called the goblet cell. This is more than likely respiratory, okay? And these cells here, these funny looking things, these are goblet cells. I'm going to talk about them shortly, all right? So this is almost a dead giveaway. If you see cilia and you see these goblet cells, these sort of things that look kind of blown up, that's a dead giveaway for respiratory. Okay. Now here on this side, you have a simple columnar epithelium. All right. So you can see that these are all these cells are attached. You can see the nuclei are all pretty much in the same layer. You've got goblet cells again. All right. So this to me is a dead giveaway for digestive. All right. And I actually know that this is digestive. I can tell. All right, I didn't pull these slides out. A few of my slides I have from Dr. Tice because I covered for her one spring. So this was just convenient. But I can tell you, I know this is digestive. You can check with her. I just know this because I can tell the difference. And it comes with practice. I'm not expecting you to go, oh, this is digestive, Dr. Bora. I'm not expecting you to do that, right? I actually studied gut for a while. So I'm just sharing with you how I can tell the difference, all right? So here you can see goblet cells in each case. This one is ciliated and pseudostratified columnar, so I know that this is respiratory. This is simple columnar with goblet cells, so I know this is digestive. And in fact, I know this is through a structure called a villus. And we'll talk about, actually, you'll learn about the villus in AMP2 when you do the digestive system, okay? Okay. Now here you have a stratified squamous and a stratified cuboidal, all right? And this is tough because I understand this looks very much like a pseudostratified, okay? But the difference is that I can see that there's layers of these nuclei. So I know that they're probably a stratified cuboidal. And this is not very common. I think there's just one example of this. I think it's in the kidney. I'm trying to remember where. Oh, it could be in the testis. That might be where it is. So this is kind of unusual. Usually cuboidal is simple, all right? But this I just kind of know from experience. And this is tough. This is not something that I would ask you on a lab test, but this is what I'm sharing with you. This one is pretty obvious. You can tell this is you can see here. This is like these tiny, tiny flat cells in many, many layers. This is skin, all right? Well, you know, you'll learn this. You're going to see this in lab. But a stratified squamous is usually skin or esophagus. Since it says here that there's a lot of dead cells, it's probably skin. This is probably esophagus, if these are supposed to be different. But yeah, so here you can see this is the sole of the foot. All right, so those are the differences there, okay? And here's your transitional. I knew I had a picture of that. So here, remember I said it looks like a hot mess? Yeah, this is a hot mess, all right? So if it looks like crazy business, it's transitional, all right? So you can compare that to a stratified columnar, which also is unusual. I can't even think of where you're going to see a stratified columnar. I'm just trying to think. Yeah, I can't even think of it. If you Google it, if you do the Google, you'll find out. You can let me know. That would be nice. Somebody do the Google and let me know where you'll typically find a stratified columnar epithelia. Okay, because right now it's not coming to me. All right. All right. So hopefully that's good. Okay. Now, if you're looking at epithelia, all right, there's different types of them. All right. So now I'm looking at page 5, right? So I said that they're ciliated, right? So I said refer to slide 15. So ciliated, right? So here you got these cilia. And in general, where you're going to see this is where you've got a sort of, you need the cilia, because remember cilia are moving. So they tend to beat and create movement of fluids. And in this case, like I said, I'm dead sure that this is respiratory. So what's happening here is that they're beating the mucus, and the mucus is trapping stuff there. And this is where you're going to find this. And then there's a protective type of epithelium. And then I said, see slide 16, and that refers to skin, right? So here you've got many stacked layers where the cells are constantly being replaced. So you're going to see this esophagus or skin, right? And then there's transporting epithelia, all right? So transporting epithelia, these are going to selectively move substances between the lumen or the space and the extracellular fluid, right? And these types of things, like here, this would be, whoops, let's go back over here. So this would be an example. I told you this is gut, right? I know this. This is a villus. And there's going to be microvilli on the top here. You can't, it's not really, it's kind of hard to see them here. But what's happening here is that the material in the digestive system has to be absorbed by the blood vessels that are going to be here. So this would be some sort of transporting epithelium, all right? And what I mean by that is something like this, all right? So you've got to move things from one side to the other, and this is all, always going to be simple, all right? Not the process, but the layers, all right? So you've got to move things from one side to the other. These things can go through the cells, and we talked about the different transporters that could be involved here. Sometimes it can go between the cells, so there's a paracellular kind of thing, but usually it goes this way, right, through the cells themselves. And we say that these epithelia are polarized, so they have a definite sidedness to them, right? So they've got one side where they're attached to the basement membrane, and the other side is this lumen, okay? And there's going to be different proteins on different sides, all right? So these are polarized epithelia, all right? And it's very important to their function, okay? And the secretory epithelia, that's the fourth type, those ones are going to be releasing substances. And very often secretory epithelium can also be transporting epithelia, right? So remember back here, I said to you, I know that this is gut and there's going to be transporting here, but I also said that these cells here, these are goblet cells, they're going to be secreting some sort of mucus, right? So in this case, it's also going to be a secretory epithelium, all right? So what do I mean by that? Well, secretory epithelium are going to be producing some sort of substance that's going to be secreted out into the surface or onto the surface or into the lumen. And these situations can occur in different ways. You can have individual cells in the epithelium that are going to produce what's being secreted. Sometimes they produce enzymes that are needed, so for the digestive system, or they're producing, say, mucus, like in this case, right? So these are these mucus-secreting cells that are referred to as goblet cells, okay? So these are called, so these cells are on these structures called mucous membranes, all right? So this is where it gets weird because a mucous membrane is always spelled M-U-C-U-S, all right? But the mucus, all right, can be spelled M-U-C-U-S or M-U-C-O-U-S, all right? So I try to be specific by saying a mucous membrane, right, secretes mucus with O-U-S. But you can see definitely that you're going to find some books that will always spell mucus and the mucous membrane the same way, okay? Just so you know, all right, both are going to be correct. I'm not going to mark it wrong, all right, just so that you know this, okay? Yeah, so it will depend, because it depends on which book you're using or what source you're using on the internet, okay? Now, and then some of them also secrete hormones, right? But these would be released into the blood. So that's going to be a very different type of situation, all right? That isn't usually what we're thinking about when we're talking about an epithelium, all right? So it would be glandular structure. I'm going to talk a little bit more about that to help ground you in this concept, all right? Now, so when we're secreting from single cells onto surfaces, we say that these are single cell glands, or you can have glandular structures, all right? And these glandular structures are classified into different ways. So these exocrine glands, so exocrine meaning that it's not going into the blood, right? Endocrine glands would be secreting into blood. So what we're referring to mostly here are exocrine glands, which could be single cells, or they could be these glandular structures, all right? And there's a way of classifying these, so you can classify them in different ways. You can classify these exocrine glands by their structure. So simple, meaning that there's, you know, no branching that's taking place here, or compound, where you've got branching. Then you go by the shape of the glandular structure. So this part here is the duct. You can see it here a bit better. So here's the duct, and then where the cells are actually producing, that's the gland. Okay, so the different types of cells, and then there's a duct coming out of there, right? So here you've got these ducts going out onto these surfaces, and the glandular cells, the ones that are doing the secretion, they could be found in a tubular structure or in what we call a sinus. If you had this in 3 dimensions, it would look like a ball. And then, of course, there's got to be something that to, you know, make that even more complicated. You could call them tubular acinar. So these sort of like elongated balls. So they're sort of in between tubular and acinar. But compound, notice that they always have these branches, all right? And then you can also classify them by what they're secreting. So serous, remember when we were talking about our serous membranes, they're secreting some sort of watery fluid. Mucus, so a mucous membrane, M-U-C-U-S, is secreting mucus, M-O-C-O-U-S, right? Or sometimes U-S. Mixed, so they're going to be secreting all kinds of stuff. Cytogenic, this sounds weird, but cytogenic glands secrete cells. So what kinds of cells, glands would those be? Well, ovary and testis, right? So there's, that's your example. So the ovary and the testis are considered glandular structures, not only because they're producing hormones, but they're also secreting their cells. So ovary and testis, right? Now, there's different ways that exocrine glands can secrete their secretions. So merocrine, which is also called eccrine, their substances are released by exocytosis. So the vesicles will fuse with the apical part of the cell membrane and open up and deposit that into the space. The holocrine, the entire cell falls apart. Okay, so the whole cell just sort of breaks down, and you know, you'll get pieces of cell as well as a secretion. Apocrine, of course, is going to be in between the two. So it's mostly kind of like a merocrine secretion, but they could also secrete these, like, I hesitate to call them vesicles because it would be confusing, but it's sort of like a membrane-bound structure that will be released, but it doesn't cause the death of the cell, okay? And so there's very specific examples of each, but most of the time, the secretions are merocrine, okay? Which should make sense because then you're not destroying a cell and you don't need to replace it, all right? Or you have to repair a cell. Okay, so hopefully that makes a bit of sense to you. Okay, now remember I said to you exocrine and endocrine, and both of them are glandular structures when we're having like a multicellular gland. And each of them are formed from an epithelium, right? So you start off with this epithelium, and then the epithelial cells, this is during development, sort of like, you know, dig into the connective tissue. And then if they form a structure with a duct, it's an exocrine gland, it's going to go onto the surface. But the endocrine gland, it will lose its connection to the epithelium. And it actually, the cells here will start releasing types of factors that will bring in a blood supply, right? So this is called angiogenesis. And then they'll put their secretions into the bloodstream. So that then becomes an endocrine gland, which is not usually what we're talking about when we're talking about an epithelium, because of this, you know, the loss of connection to the original epithelium when this was made, or when it was formed during development, okay, or embryogenesis. And you'll have a whole chapter on the endocrine system after the break, so when you start in the spring semester, okay. Okay, so now what are your types of secretory epithelia? All right, so again, they're going to be secreting. You've got all these different types of secretion. So they're going to form these different types of membranes. So here you have your mucous membrane, right? And again, this can also be spelt with just the U, all right? So again, it's going to depend on your book. right? But I'm trying to keep straight in my convention, and you'll see that in my notes. So a mucous membrane with just the U secretes mucus with OUS, all right? So I'm just being clear on the way I do it, and that's usually the way I see it. But you can see that it will be switched around depending on the book and where you find your materials, all right? So I want you to be really pretty adept at that, but don't let it freak you out, okay? So mucous membranes secrete mucus. Then you have serous membranes. They're going to be secreting that sort of liquidy material, so watery material, right? So here, think of your serous membranes that we talked about, your double membrane system around your lungs, your heart, and your gut. So your pleural membranes, your pericardial, and the peritoneal membranes. Right? So those would be your classic serous membranes. Then we also have synovial membranes that we'll talk about later in the semester. Those are going to be found in your fluid-filled movable joints. So they're going to be secreting synovial fluid. And then your cutaneous membrane, which is your skin, right? So your skin secretes all kinds of stuff, right? So sweat and oils and tears, right? So that would be your cutaneous membrane. Okay, so then here I've got a few types of epithelia from, and this would be more like what I would do in lab. I think this is pretty clear. I'm not out to make it really hard on you, okay? So look at these and see if you can identify what types of epithelia we have, right? So here's one, and then here's two, and then here's number three, okay? It's not that you have to do it, but if you want to know if you're right, you can send me an email. You don't have to, all right? But if you want to know if you're right, send me an email and tell me what you think they are, okay? Okay, now I said to you that these epithelia have to be connected, right? They have to be connected to each other and to the basement membrane, all right? And there's different types of junctions that are associated with that. So I have a couple of slides here that go over these, all right? So I'm going to talk about it on this slide, and then I'll move to the next slide, right? Because it's hard to find one, picture that has everything in it that I want. And here, this is actually from the journal Nature. If you want to find this, sometimes I put my sources for my images on here, but this was a nice review of different types of connections, and you can, if you're interested in this, you can follow this link. You'll go to this review paper and you'll see it there. But anyway, so here again you have your apical surface and you've got your basal surface, and here you have your extracellular matrix. Know that this is your basement membrane, all right? And you've got the different types of junctions, all right? So and then we're going to start talking a little bit about their connection to the cytoskeleton, right? And to the extracellular matrix, all right? So notice that we have different labels here, right? And so if you look at my notes, I'm very clear on what these things are connected to. So the first one are these tight junctions. They're usually found towards the apical surface. And these very often, they're called occluding junctions sometimes, and that means that they're making a seal. All right, and not every epithelium has these, but think of where you might have it. So maybe the gut, for example, someplace where there's sort of something fluidy on the inside. All right, you won't find it in blood vessels because the blood vessels are simple squamous. So you don't have enough thickness to form these. All right, except when you're talking about the blood-brain barrier, but that's special. So we'll talk about it later. All right. Now, these tight junctions, and there's a number of proteins that make these junctions. Now, these will form a tight seal. You can see in my notes, and they're connects the cells to each other, but also to the cytoskeleton, specifically to actin. All right. So that you can see here. The next ones are adherens junctions. All right. So these are cell-cell. All right. And these are going to connect to the actin cytoskeleton, all right? Now, here, in the particular type of proteins that are found in the membrane here where they're touching each other, these molecules are called adherens, all right? And just so you know, they're called adherens. And then the next type of junction is called the desmosome. Now, these desmosomes are also cell-cell connectors, but they don't connect to actin. they connect instead to intermediate filaments, all right? And these ones are going to be found in spots or patches, whereas the adherens junctions, they actually form a continuous belt around the cell, all right? So you don't get that perspective from this picture, all right? Now, there's also connections to the basement membrane. So here we're going to have cell to the extracellular matrix. The first one would be hemidesmosomes in my notes, okay? So here what you have, again, connections to intermediate filaments. And then instead of being via adherens, you've got integrins here, okay? Now, the other ones here are the focal adhesions. So these are also cell extracellular matrix. And what they're forming here is a connection to actin. And down here, you've got integrins as well. So in each of these cases, you have integrins, all right, connecting to different aspects of the cytoskeleton. Now, how are they different? Well, these ones, the focal adhesions, they allow for cell motility. All right, so you don't typically find them in an epithelium unless you're talking about development or if there's some sort of disease process taking place. Okay, in development, some of these epithelial cells are moving around, so they're going to have a different type of connection to allow that. Okay, okay, so just saying on the bottom, it's integrins, adherens junctions. You've got adherens in there. All right. Now, if you look at the adherens, junctions, and the desmosomes, their function is to prevent the cells from separating, right? Whereas with the hemidesmosomes, it's helping to anchor the cells onto the basement membrane, right? Whereas again, focal adhesions, those are for motility. Now, so here's another look at that. So you got your tight junctions, adherent junctions. This one here, you can see the belt, the adhesion belt a little bit better. That's why now I've added this one, but you don't see the connection to the cytoskeleton here, which is the part I don't like. So like I said, it's hard to find one picture that has everything in it that I like. Okay, so yeah, and then here's another perspective here as well. So hopefully, you know, all of these together will give you a good appreciation for the types of junctions, all right? Now, the next few slides, now this is just to give you a bit more of perspective on the importance of this, all right? It's not like I'm going to be putting this on an exam. I'm not. But I want you to appreciate what's happening here. So, because we're going to talk a little bit more about it as we go on in the presentation. So I wanted to seed you here. So there's this thing called the epithelial mesenchymal transition, so EMT, all right? So not emergency medical technician in this case, epithelial mesenchymal transition. And to introduce this, I have this idea here to let you know again about the extracellular matrix. Like earlier on in the semester, I was extolling the virtues of the cytoskeleton and all the different types of proteins that are there and what they're used for. And you could sort of see that here with these junctions, right? Different junctions to actin or intermediate filaments. Notice that the microtubules are not involved in this, right? Microtubules are cytoskeletal components that are not involved in these adhesive processes, all right? So that's different, right? But what I want you to see here is that the extracellular matrix as well is really important in terms of what is there, right? So I sort of alluded to that back here with the basement membrane, right? So I showed you the basement membrane isn't just, proteins. There's like a highly organized and very specific proteins involved here, okay? So when you're looking at the extracellular matrix, which is very important here, I want you to sort of see there's sort of, in general, three classes of proteins, all right, that are involved. So first of all, you have the structure. So the structure, two very common types of proteins there are collagen and elastin. Now, I already showed you there's many different kinds of collagen, all right, but just say collagen and elastin, they're responsible for the structure. All right. Then you have proteoglycans. So proteoglycans mean that there's more sugar, more protein than there is sugar. All right. So it's mostly protein, and then the glycans is some sugar. Okay. And I listed an example here. One is called fibromodulin. All right. But basically what these proteoglycans are doing is that they're stabilizing the structural proteins, right? So the collagen and the elastin in a particular form. And then we have glycoproteins. So that's going to be the opposite. So they've got more sugar than they have protein. And they're really helpful to allow the cells to attach to the matrix, okay? And here you have fibronectin and laminin, all right? I'm going to talk a little bit later about fibronectin. So this is just to give you an appreciation that there's very different types of proteins here and in their general functions, and understand that there's going to be different types of laminins, different types of elastin, different types of collagen. So if you're someone who studies this, you can gain an appreciation of it. For myself, I used to study cell motility, so I knew that I was aware of this, but I wasn't, well versed in exactly every type of collagen, right? For example, again, very, very specialized kind of work, all right? But the reason I'm mentioning this is because of this epithelial mesenchymal transition. And here, if you're interested in this, you can follow this. It's going to take you to where I got this image, but this is from a journal called the Journal of Cell Science. And what it's teaching you, so they're using epithelial mesenchymal transition during development. And we were already started talking about how these cells move around and they change. And if you watch the video, you'll see that there's all this signaling taking place. So here's gastrulation. And after gastrulation, you start getting this thing called the neural crest. And you'll see that in the video. But this is really important because the way the cells know where to go, they know where to go because of the extracellular matrix that's being laid down, all right? And the cells are moving, like I had said to you, okay? So sometimes they move as individual cells, and sometimes they move as sheets, which is really interesting, okay? So you can have this epithelium. So epithelia are very static systems, linings and coverings. But sometimes what will happen is that you'll lose the dissolution of the cell junctions. The cytoskeleton is going to rearrange itself, and you're going to get increased motility, all right? So this is normal during development, but this is also what happens during the spread of cancer, so invasion and metastasis, all right? So the cells will lose their cues, and this is sort of telling you, what types of genes, and there's like all of these different things here. I used to study this one here, okay? In terms of how the cells transition from a normal epithelium to a cancer type of situation, and the cells will start to move around and invade. And that's because their connection to the extracellular matrix becomes changed for some reason, all right? Now, a really cool example of this, there's this scientist, her name is Mina Bissell, and here's a TED talk from her from a few years ago, maybe could be 5 or 6 years ago, but she's amazing to listen to. So for some reason, the hot link doesn't work, but if you want to learn about this, copy and paste this link, you get to her TED talk, and she's really funny. I had a friend who was working in her lab as a postdoc. But basically, she was one of the main people that was bringing out this idea that the context in which the cells are existing in is really important to their function. And she, of course, was studying breast cancer. But in this talk, she's going to talk about how these breast cells would change from normal to more of a cancer type of phenotype based upon what genes were turned on and how those cells were attached to the cell matrix. Okay, so basically, if there was one gene involved, if it was turned on, the epithelial cells would form glands and start secreting milk. But if that gene was turned off and other genes were turned on, those cells would then start to move around and invade. All right, so it's really interesting how this works. And I just want you to get an appreciation of this. Again, I'm not going to put it on the exam, right? But this is just to generate your interest in the importance of how this works, okay? Okay, now there's one other kind of junction that we're going to talk about in a little bit that doesn't tend to be found in epithelia, all right? Unless those epithelial cells become cancerous, sometimes you'll find this junction. These are called gap junctions. All right. Now, normally, where do you find these cells? These junctions. Normally, they're found between types of muscle cells. So we're going to be talking about muscle tissue in a little bit, but since I'm talking about junctions, I'm introducing this here. So gap junctions are very often called communicating junctions, and they're cell-cell junctions. Okay. So you can see a gap junction is made up of one half on one cell, and one half on the other cell. And this half is called a connexon. And each connexon is made up of 6 connexin units. And 2 connexons, one from one cell and one from another cell, join together to form a gap junction. And this is basically a hole between 2 cells. So small molecules and ions can actually move between these cells. And the cells can communicate to each other directly. All right. You'll see how important that is for cardiac muscle later on in the semester. Now, these junctions can be closed as well. They don't always have to be open. All right. So they can be opened and closed. All right. So, so those are gap junctions. And again, communicating junctions. All right. And we'll talk about them shortly. Now, connective tissues. Connective tissues sound crazy. There's a video at the end of this that you can watch to try and bring this together, but it can sound to a student that this is just a hodgepodge of stuff, okay? But it's not, and it's because they have a common embryological origin, all right? So they're grouped together because of that, all right? So embryonically, they come from a connective tissue called the mesenchyme. And this is needed to form all of these connective tissues. And it's sort of a mesh of proteins with a fluid matrix, right? And most of that mesenchyme comes from the mesoderm, most of it. And a little bit comes from a specific part of the ectoderm, which is the neural crest. And that forms after gastrulation, all right? So you'll see that. So I'm getting into this here. The embryonic connective tissues, all right? So this is where I'm talking about this. So the 2 embryonic connective tissues are the mesenchyme, and that's what I'm saying gives rise to all of these connective tissues, all right? So you can see it here. And like I said, right, mostly from the mesoderm, all right? A little bit from the ectoderm, specifically neural crest. Now, we also have this mucous connective tissue, but that's not actually found in the embryo. It's found in the spinal cord. Sometimes it's called Wharton's jelly. And that's where all of those stem cells are. So if you have a baby and you bank the stem cells, that's where they're getting them from, right? So they're getting them from that Wharton's jelly in the umbilical cord, all right? So embryonic connective tissues. Now, mostly, however, we're going to be talking about mature connective tissues. Now, this first set of slides, all right, these are going to really sound like connective materials to you, all right? Because there's fibers involved, right? You're going to be thinking of connective tissue fibers, right? So these types of sort of fibrous-y, lining support tissues are either called loose or dense, all right? So I'm on page 11 of my notes, okay? So loose versus dense, what's the difference? And if they're relative to each other, okay? So this sounds really weird, but we say loose are flexible, but not as strong. And dense are strong, but not as flexible, okay? And you'll see that under loose and dense, we have 3 categories in each one, all right? So the first one under loose is areolar. And the reason we're starting with areolar is that most loose connective tissues are areolar. So if you're in doubt, just say areolar, because you're probably going to be right. And you definitely tell it apart from adipose, which I'll show you in a sec, all right? So most loose connective tissue is areolar. So it's widely distributed, has several different types of cells and fibers, and it's used to attach things like, to other things, right? So skin to the underlying tissues, and it's also used to pack. That's just going to be between glands, muscles, and nerves. So if you see muscle cells, and then you see what kind of connective tissue, you're going to see areolar, right? If you're going to see nervous tissue, and it's going to say what kind of connective tissue is here, you'll say areolar, all right? So most of the loose connective tissues are areolar, all right? Next to that, we have adipose. So adipose Adipose is fatty, right? So it's storing your triglycerides, all right? Where are you going to find this? Well, it's subcutaneous, right? It looks like you'll see these big open spaces, all right? It looks almost like there's air, and it's because when it's sectioned, the fat comes out. Sometimes you'll see it's sort of yellowy if the fat stays in there, all right? Now, what is it for? Well, it's going to be insulation and padding, and because it's got fat in it, right, energy source, all right? Now, the last one, this is the hardest one to remember, is reticular connective tissue, all right? Now, where do you find it? Now, this is very specific. It's again, forming a scaffold. It has these things called reticular fibers. Very often they look kind of thick, or thicker, not as thick as collagen. But you'll really find it in lymphatic tissue, right? So you're going to find it in the spleen and the lymph nodes, right? So this is a lymph node here, so you can see that there. So it's kind of specific to that, and I don't really know why that is, but you know, hopefully that will help you a little bit. So hopefully you could definitely tell the difference between areolar and adipose. This is a little harder, depending on the staining, right? But if you see a slide of lymph node and it's, you know, what kind of connective tissue here, you'll say that it is reticular, all right? Now, dense also comes in 3 different flavors. So you have dense, irregular, all right? So right away you can see this pink, this is collagen. And generally these fat, pink fibers is collagen. And here, notice that there's no rhyme or reason to the pattern here, okay? So let me say it's irregular. All right, so this provides strength when forces are pulling from different directions. Now, dense regular, all right, looks very different. Now, again, you see these collagen fibers, and notice they're all aligned with each other, all right? So this is where you're going to find it, tendons and ligaments. You're going to find it in where you're going to have a directional pull, all right? So one axis. And then there's elastic. So elastic allows for the expansion and contraction of tissues, all right? So you'll find it here, you can see it, it's in the aorta. You'll see it in, you know, where your ear is as well. There's think that that's going to be collagen, but you're going to see it here in the aorta. Stretching of elastic arteries is a very common one. Okay, so here you can sort of see, compare and contrast these. So here you can see loose connective tissue in different cases. So here you can see your lipid in your adipose. Here you can see some reticular fibers. Sometimes they'll help you by staining the reticular fibers. They'll appear sort of black, but that can be confusing because elastin can also be stained black, all right? If you see it in a case like this, the reticular is sort of the fainter ones, and then you can see the collagen usually tend to be a little bit thicker, and the elastic are the darker ones, all right? It's a little bit harder to tell, all right? And I know that, right? But this shows you the difference between areolar, reticular, and adipose, okay? Here, dense, regular, and irregular. Hopefully that looks pretty obvious, which one's regular and which one's irregular. Now, cartilage, all right? Now, there's 3 kinds of cartilage, and I sort of put these slides here together. You have fibrocartilage, hyaline cartilage, and elastic cartilage, all right? So this now is getting us into specialized connective tissues. Now, this is where it gets really weird. So we had these very specific types of, you know, sort of fibrous, sort of, you know, sort of amorphous functioning tissues. But there's also these specialized connective tissues. And again, because they come from the same, you know, origin, right? So cartilage is one of them. Now, cartilage, all 3 cartilages, the cells are called chondrocytes, and the cells that form cartilage are called chondroblasts. So chondro means cartilage, all right? Cyte means cell, and a blast is an early type of cell, all right? So here you've got these 3 types of cartilage. So hyaline cartilage, elastic cartilage, and fibrocartilage, all right? So on page 11 of my notes, you'll see hyaline cartilage, in general, we say it's smooth and glassy. It's mostly collagen fibers, all right? So you can see it's very sort of smooth looking. These are the chondrocytes in the middle here. And this is the most common type because it's found on the ends of all of our bones. And it's very important for the movable joints, right? Because those is a very smooth cartilage. The fibrocartilage, now if you look at it, it doesn't have that sort of smooth look to it, right? You can sort of see fibers in here. So the collagen bands are thicker. Now this is a very strong cartilage, and you'll find it in your intervertebral discs. And also, if you damage your hyaline cartilage, you can't replace it. And there's a reason for that, which hopefully I'll talk about when we do joints and bone, all right? But the hyaline cartilage gets replaced with fibrocartilage, which is why if you damage a disc or if you damaged the hyaline cartilage in a joint, afterwards your joint doesn't bend sometimes doesn't bend as smoothly, because wherever you damage your hyaline cartilage, you're going to replace it with fibrocartilage. And then you also have elastic cartilage. So this is going to have elastin in it, and this is a flexible cartilage. This is going to find in the end of your nose and the pinna of your ear. Okay, so it's collagen and elastin. All right. Now, here you can go walk you through the notes here. So cartilage is a tissue with very poor blood supply, grows slowly. All right. So hyaline cartilage, the most abundant type of cartilage, the ends of your bones. The fibrocartilage, again, thicker bundles of collagen fibers, very strong. And then, of course, you also have your elastic cartilage, okay? The elastic cartilage, of course, is going to be elastic, so it's flexible. Now here you can see a different look at this, all right? So this is how you're usually going to see it in lab. So the smooth, glassy appearance that you have here on the hyaline cartilage. And the elastic cartilage, you can actually see the elastic fibers in here. And then notice with the hyaline, with the fibrocartilage versus the hyaline cartilage, again, you can see those collagen fibers, all right? And then, of course, the elastic cartilage, right? It has your elastic fibers. So here's your full set of notes. Okay, now, bone is another type of mature connective tissue. So here what you're going to find is a hardened matrix, all right? The cells here now are called osteocytes. So osteo means bone, and then cytes is cells. Now the cells that form bone are called osteoblasts. And you'll see that there's two types of bone. So there's spongy and compact, all right? And you'll see that, and we're going to go over these in more detail when we do bone, so that you kind of learn the structure there. But the bone is sort of arranged in these structures called osteons, and you can sort of see that, you know, this kind of idea here. You can see canaliculi and lacuna, and we're going to go over this structure later in the semester when we talk about bone, okay? But you can sort of see these sort of structures here that we would call the osteon. And you don't need to learn the osteons yet. We will learn about them later, all right? But here you have your compact versus your spongy bone. The spongy bone is sort of towards the middle of your bone. It's sort of a loose kind of structure. It's covered with the compact bone. This is much denser. We'll talk about what chondroitin sulfate is. It's the mineralized matrix that you have in your bone. Calcium, obviously, you probably know that's one of the minerals there, but also phosphate is also in there as well. And like I said, we'll talk more about this structure later when we get into bone. The other term for bone is also osseous, right? So that's why you have an osteocyte, so it's osseous tissue. You might see that in some of the books that you're looking at. Now, blood and lymph, again, that's another type of connective tissue. So you're gonna have a whole chapter on blood next semester. So when you're talking about blood, you've got many different types of cells, and each of them have very specific functions. The cells are very motile. The matrix itself is also very fluid. It's called plasma, right? That's what makes up the most of your blood. You're going to learn different components of plasma later on, as well as what you have lymph for. So you're going to learn much more about that in semester 2, but it is a type of connective tissue. And then, ironically, adipose you'll see as well that in addition to it being a loose connective tissue, sometimes it's also referred to as a specialized connective tissue, all right? So in this case, you've got very few fibers and many cells. So this is why sometimes it's a specialized connective tissue as opposed to, you know, a loose connective tissue, because, you know, those other tissues, it's mostly fibers and fewer cells. So this is why adipose sometimes appears as a specialized connective tissue as well. And again, it's going to store your triglycerides. Now here's a good review, I think it's a crash course review of these different types of connective tissues. And you'll see when you go to that link, there's a whole bunch of other videos to other different types of tissues as well, right? But this specific one is to connective, all right? So hopefully that's helpful to you. Now, muscle and nervous tissue, all right? Here you can see the cells are, the types of cells are different, but then we often talk about them together because both muscle cells or fibers and neurons are excitable, all right? So what does that mean? Well, that means that they can carry an action potential, all right? And that, what is an action potential? Well, it's an electrical signal. Now, something that we didn't really talk about yet in this course is the understanding that that the cell membrane actually has a charge across it, all right? And all cells have this. And it's called the resting membrane potential. And it has to do with the different charges on the different sides of the membrane. So if you remember, I would show you these pictures of the concentration of sodium ions across the membrane, the concentration of potassium ions across the cell membrane, and calcium ions across the cell membrane. Okay, and they're not equal. And in general, this produces a charge gradient across the membrane. And this is called the resting membrane potential. And all cells have this. But it's muscle cells and neurons that are able to flip this charge or generate what we call an action potential. And we'll talk much more about this later in the semester, how this works, and the details with this. But basically, it's involving the concentration, the local concentration of sodium and potassium ions, and it causes the resting membrane potential, which is usually about -70 millivolts, to flip and become positive. And it can go up to, you know, 25, 30 to 40 millivolts. And then, so we see it depolarizes, and then it will repolarize again. So it will flip back to the negative, all right? And it's muscle cells and neurons that can do this, all right? And that's what we mean by an excitable cell. So its resting membrane potential will do a flip, okay? Yes, so why does it do that? Well, it's responding to some sort of a signal, right? So a neuron will create an action potential because it's sensing something, or it's been signaled by a sensory cell, and it will carry that information on to either another cell or a muscle cell or a gland. And whereas a muscle cell will do this in response to a nervous signal from a neuron, right? And then what will its reaction be? Well, it will contract, okay? So but both of them involve in this carrying of charge called an action potential, all right? And we'll talk much more about that in detail later. Now, if we focus specifically now on nervous tissue, all right? So when we talk about nervous tissue, we're referring to the neurons themselves, as well as the neuroglial cells or support cells, right? So nervous tissue has the neurons there, as well as the support cells, which are the glial cells. Sometimes they're also called accessory cells, all right? So there's many types of these glial cells that are really important for function. So neurons need those cells in order to do their job, all right? Now, if you look at a neuron, all right, they have a directional movement of the action potential. So it starts at the dendrites, and it's transmitted through the cell body down the axon, and it eventually gets to this structure called the terminal end bulb or plates, and then passes that to a second cell. So that second cell could be another neuron, it could be a muscle cell or fiber, or a gland, okay? And then there's going to be some sort of response to that. Now, the accessory cells, there's going to be different types of neuroglial cells depending on whether you're talking about the central nervous system, which is the brain and spinal cord, or the peripheral nervous system, which is everything else. Okay, so the neuroglia and the central nervous system come in several forms. So we have oligodendrocytes, astrocytes, microglia, and ependymal cells. And they have very specific functions. And we'll talk, I think I'm even going to use the same slide later, and we'll talk very specifically about what those functions are. All right, but you can see we've got 4 different cell types, 4 very different functions. And then the peripheral nervous system has 2 types of glial cells, so satellite cells and Schwann cells. And again, we'll talk much more about this later in the semester, okay, and exactly what they're doing. Now, muscular tissue, right? We have 3 different types of muscle cells or fibers. The cells are often called myofibers or muscle fibers. And like I said before, they're contractile. Now, myo means muscle, and the reason they're called fibers is because very often these cells look sort of fibrous, right, in their appearance. Now, walking you through this, so skeletal muscle, this is a voluntary type of muscle cell. So what does that mean? Well, you control it consciously with your mind, right? So those types of muscle are found on your limbs, right? You can move your arms, move your legs, move your body, okay? And the commonalities here is that we say they're striated. So there's these lines on them, all right? We'll talk later what these are, but they're based on a structure called the sarcomere. And these cells are really, really long. Like some of your muscle fibers, your skeletal muscle fibers, run from your hip to the inside of your knee. Okay, so there's a muscle there called the sartorius muscle, and those cells run that entire length. Okay, so they're very, very long, and they have many nuclei in them. All right. Then cardiac muscle cells, they're similar to skeletal muscle cells in that they also have these striations. So they have this sarcomere structure, but that's pretty much where the similarity ends. Cardiac muscle, obviously you don't control that, so it's involuntary. They have only 1 nuclei, so the cells are much smaller, but the cells themselves can be sort of branched, all right? And the junctions of these cells have these special names called intercalated discs, and in those intercalated discs you can find gap junctions and desmosomal adhesions. All right, so the desmosomes are going to make sure that those cells don't pull apart. You can't, you don't want to get a stretched heart muscle. You can do that with your skeletal muscle, but don't do that with your heart. You're going to pull your heart muscle. And the gap junctions are communicating junctions. So we'll talk later how that works, but that way the ions can move directly from cell to cell and move that action potential very quickly between the cells. So obviously you find cardiac muscle in the heart. Then smooth muscle, all right, it's sometimes called visceral muscle. This as well is involuntary. Unlike the other 2 types of muscles, not striated, okay? Now, that means it doesn't have a traditional sarcomere structure, which will mean more to you later in the semester. They also have single nuclei. The cells themselves are sort of spindle-shaped. So they have sort of elongated, would come to a point, right? Think of a spinning wheel where they used to have the spindle there. Sometimes they're found in multiple layers, 2 or 3 layers, and hopefully I'll get a chance to talk more about that later. And you'll find them around tubes, so around your blood vessels, around your respiratory tract, around your digestive tract. So we're going to help move things through those tubes, right? And in blood, control blood pressure, in the vasculature. You'll also find smooth muscle at the ends of each of your hairs. There's a piloerector muscle there. So it's just smooth muscle. Important for moving your eyes as well. It's also smooth muscle. In some cases, you'll also find gap junctions here, all right? So if you have a whole bunch of smooth muscle cells that have to work together, very often there'll be a gap junction there between those cell types, or between those cells, all right? But you're not going to find intercalated discs. That's specifically cardiac, all right? Now, a few other ideas Oh, okay. So here you can see another look at skeletal, cardiac, and smooth muscle. You know, a little bit. Hopefully, you can tell which one's which. If you're at all confused here, make sure you ask. All right. And then here's a table that sort of summarizes everything what I just said. So hopefully this will help you. Now, some other terms that we need to go over in terms of tissue growth and differentiation. So these would be terms that you could refer to abnormal or normal situations. So hyperplasia, all that means is like the growth of a tissue through multiplying cells. But clearly that could go wrong, in which case then you get neoplasia, and that would be the abnormal growth of a tissue by hyperplasia, and you can get a tumor form. Then you can have hypertrophy, which would be the enlargement of cells. That can happen in a weak heart. The muscle cells will enlarge, but it can also happen normally if you work out. Your muscle fibers will, you know, get bigger. You don't get more muscle cells. The muscle cells get bigger. Then you have this idea of differentiation. So the cell will gradually develop into a more specialized form and function. I'll show you an example of that. Then you can have metaplasia, in which the cells change from one mature cell type to another. All right, so let's think about that a little bit here. So here's a classic slide that I use for the development of cancer. All right, so so here you can have a hyperplasia situation where you have a cell which will multiply, and this could be normal, right? It could be a normal situation, but in cancer it will also occur as a first step. But at this point you don't have a cancer, you just have like a mass of cells here, and they're normal cells. They look like they're neighboring cells. That will then go on to a dysplasia, where you'll get some cells that look weird, that don't belong there. And then you can get a benign or in situ tumor. So the tumor is localized into a particular location. And then you can go on to a malignant tumor. And this is where you start getting a neoplasia. You might see this as well, neoplasia. But here you've got this abnormal growth of weird cells. So that would be neoplasia. So always when you see that, it's sort of implying cancer. So that neoplasia. And then a metastatic tumor is where the cells actually are moving to a different place. So in a malignant tumor, the cells are like breaking free of their surroundings, and they're sort of trying to invade. And if they get to the bloodstream and then extravasate or leave the bloodstream in a secondary site, that would be a metastasis. All right. Anyhow, so there's some other terms for you. Now, so here's examples of hypertrophy. So here you can see your muscle cells are getting bigger. You're not getting more muscle cells. You're getting bigger muscle cells. Metaplasia. Again, here you can see an example where you're getting a conversion from one cell type to another. And I have another slide where we talk a little bit more about that. Differentiation. So you have, usually this is where you have some sort of a stem cell. I'm going to talk about stem cells, but they get a signal that you need to make more of those. So remember where I was talking about osteoblasts and osteocytes. So a blast cell then forms bone. So the blast would be sort of like a stem cell, and that's going to go through a differentiation process to form the osteocyte. So that's what you're seeing here. And you can have the same thing for collagen formation. You'll see it as well for blood formation as well. So you're going to have a cell that's going to it's going to divide, but it's also going to differentiate as it divides and become more mature, right? So you're going to differentiate. Here's my slide on metaplasia. So this is kind of a weird thing. Now, importantly, when you're having one type of cell convert to another, you're going to lose function, all right? And here, if you're interested, you can follow this link and listen to this guy talk about, his experiences. He's an expert on metaplasia. But when would you have this? Well, toxins due to cigarette smoke can cause metaplasia of the bronchi. So all of a sudden, you're not going to be able to breathe as well, and you won't be able to transport gases as well. A bacterial infection of the digestive tract can cause the intestinal lining to switch to a squamous type. So That can also occur. So you can see here with smokers, you can get your pseudo-stratified ciliated respiratory epithelium becomes a stratified squamous. And this is, stratified squamous is like skin, right? So it's a protective thing, and you're not going to be able to get gases through here, all right? So this is sort of the importance of metaplasia. Now, in terms of stem cells, so we've got different types. So not all stem cells are the same. And you probably realize this if you're listening to, like, what, stem cell therapies and what you can do with them. So when you're talking about embryonic stem cells, you have totipotent stem cells, which basically the first cells are totipotent. So they can form any type of tissue. And then later on, you're going to have pluripotent, so you're going to have stem cells that can make, more than one type of tissue, but they're a little bit more limited than the totipotent. So totipotent can form anything, which is why embryonic stem cells really are the ones that one wants to use for therapies, because you can push those stem cells to form all kinds of different types of adult tissues. Now, in adults, the most you're going to get is a multipotent stem cell. So you can have a stem cell that can form maybe a couple of different types of tissues, but most of them are unipotent. So where I say, an osteoblast, well, it's a stem cell that forms, bone, right? So, so those are unipotent. So they can only form one mature cell type. So here you can see this sort of idea. So totipotent can form everything, including pluripotent embryonic stem cells. Pluripotent can form different types of tissues. So, each of these, the endoderm, mesoderm, and ectoderm, those stem cells can form many different types of tissues, clearly, because there's many different types of tissues that arise from each of those germlines, right? Multipotent, they're a little bit less in their capacity, right? But they are still stem cells. The interesting thing about adult bone marrow and skin cells, as well as cord blood, those multipotent, sometimes you can put them in the lab and you can bring them back down to pluripotent, which is why when they're looking at adult stem cells for therapies, very often they want to use bone marrow cells, because they tend to be able to do this. It's kind of neat. They can push them back to pluripotent, which is very useful if you're only using adult stem cells. And then, of course, you can have unipotent, which is what I was just talking about. So you'll see that there's stem cells in skin that will only form keratinocytes, for example, and so on. All right. So, we will talk more about these ideas later in the course, or when you get into semester 2. Now, the other thing is healing of tissues. So this is a really big idea, but basically there's 2 ways to repair tissues. One is complete regeneration. If you're regenerating tissue, you're going to completely reform the tissue as it was before. And it's very complex mixing of stem cells and signaling that's going on there. The organisms I study in the lab, planarian, they're notorious for regenerating. You can cut them up into theoretically 200 different pieces and get to 200 new planarian. So they're really, really great regenerators. Fibrosis is when a tissue heals, but it doesn't completely reform like it was before. So remember I had said to you that if you damage your hyaline cartilage, you'll get cartilage back, but you can't get your hyaline cartilage back. You get fibrocartilage instead, okay? All right, now, so here's an example. And actually, your liver as an adult, your liver has a high regenerative capacity, but I can't get a good picture of regenerating liver. But the very tip of your finger, this might sound really bizarre, but if you lose the tip of your finger, if you just nip it at the very end, your finger can regenerate to an extent, all right? Whereas fibrosis, like I said, it leaves a scar. So depending on how bad the wound is, the bigger the scar, all right? Now, there's a few other terms, and we're on the last page. So we've got a few other terms. So atrophy, that term, what that means is sort of the reverse of hypertrophy. So think of muscle wasting. So muscle cells will shrink from lack of use, right? So say you're wearing a cast for a really long time. Someone who has paralysis, their muscle will atrophy, get smaller. Bedridden patients, that will happen as well. The term apoptosis, it's actually pronounced apoptosis, it's Greek, but you'll hear people say apoptosis. I cringe when I hear that, but it's apoptosis. So what's happening here is a controlled cell death, and this is a signal that comes from inside of the cell. So the cell self-destructs, and it follows it. Sometimes this is called programmed cell death. So there's actual genes that get turned on, and this is a way we get rid of damaged and worn out cells, right? So this is a normal process. Compare that to necrosis. So here you're also having cell death. You can have, but this is an outside force causing, not that the, apoptosis, it's the cell responding to outside forces, but it's something that starts from within the cell in response to that. Whereas necrosis, this is when, something on the outside of the cell literally causes the tissue to die. So this could be some sort of diseased tissue, infections, that kind of thing. And there's a destruction of the tissue. The cells will break open and release a lot of factors that cause more necrosis. So we say that necrosis is passive because the cells aren't saying to themselves, okay, it's time for me to die. It's that they're being damaged, so it's called passive, whereas apoptosis is an active process. Gangrene is where necrosis gets dangerous, right? So this is life-threatening, right? So a considerable mass of tissue dies, and this can involve a bacterial infection, but it doesn't have to. And you have a complete inadequate blood supply. My aunt had gangrene. She lost her leg due to that, but it wasn't because of an infection. She had really poor circulation, and her tissues just started dying because they weren't getting enough of a blood supply. Which brings me to infarction. All right, she didn't have an infarction, she just had a very poor circulation. But an infarction, that can also cause, say, gangrene or necrosis, because what you have is a blockage of the blood supply, and it can cause the death of the tissue. So, for example, a myocardial infarction is a blockage of a vessel in the heart, and that causes the death of some of the heart tissue, leading to heart attack. And yes, of course, could lead to death. All right. So, so these are these different terms. All right. All right. So that brings me to the end of that idea. And now, walking you guys through this, there's a few slides that I'm going to change the orientation of, just because I think it would flow better. So I'm going to include that in the video, so that when you go back and you look at my notes, you don't, think, what the hell's going on? But just going through it, because this is a new presentation for me. So I just felt like going through it. Some of the slides weren't really where I wanted them. So you will see I'm about to move them around, and I'm going to show that to you. And you'll probably agree with me, because you probably heard me go, that sounds kind of weird, while I was talking about it. And it really came up here where we got into the cartilage. I think what I should do is I'm going to move this slide before that. All right, so we're going to go over the different cartilages, just so that you know, and you're not surprised when you see this, okay? When you're looking at it in my notes. There, I'm going to keep that there, and I'm going to keep this one, elastic cartilage, there. And then you'll have the two views. You'll have my view and then the other view. All right, so hopefully that doesn't throw you when you go through the video. And there was something else that I didn't like right here. I don't know what happened to the end of this slide. There we go. Let's fix that. The aorta, which matches what's down here. And that way you'll understand what happened. And when I post this, I'll say I moved around a few slides in the cartilage section so that you are not thrown by that. And then we had the bone, and that was okay, I thought. And then adipose muscle, I was okay with that. And muscle. Okay, I think that's okay now. All right, so hopefully that will help you and you don't get completely discombobulated. And I will put a little note saying that I changed the order of the collagen slides. Not the collagen slides, been talking for a long time. Over here, the cartilage slides. Okay, so hopefully that's okay with you and you feel comfortable. And as I said, if you ever have questions, please make sure that you ask. And now you kind of know the meaning of my slight joke at the beginning with the tissue box. Don't cry. Make sure you come and ask me questions, and I will set you straight, because there's no need to cry. It'll be fine. All right. So thanks a lot, and hopefully that was helpful.